Good day, everybody. Welcome to the webinar on early blight and brown spot of potatoes. I'm Jackie van der Walt from the University of Pretoria. During this webinar, I'm going to discuss the pathogen of these diseases, symptoms related to each of the two diseases, disease cycle, and then close with some management principles and practices. So let's start with discussing early blight. Early blight is caused by the fungus Alternaria solani. This is an aggressive pathogen and can infect plants directly. This means it doesn't necessarily need a wound or a natural opening for infection. If we look at brown spot, colloquially known as molrus, the pathogen is Alternaria alternata, although we do know now that there are other small spored Alternaria species that can also cause brown spot symptoms on potatoes. Alternaria alternata, in contrast to Alternaria solani, is a saprophyte and perhaps even a weak pathogen. Alternaria alternata requires wounds or natural openings in the plant for infection. It cannot penetrate the plant directly. To explain a little bit more about the Alternaria species, the Alternarias are necrotrophic pathogens. They secrete cell wall degrading enzymes that kill the plant cells ahead of infection, and then they live on dead plant material. So the necrotic lesions on the leaves are actually where the pathogen is sporulating, growing, and reproducing. Alternaria species have what is known as a polycyclic life cycle. This means, firstly, that they complete many life cycles or disease cycles in the growing season of the plant, but also that they produce secondary inoculum. And it's this secondary inoculum that results in epidemics. Alternaria species undergo asexual reproduction. But despite the fact that they undergo asexual reproduction, there is still a high level of genetic diversity found in populations of Alternaria species. This results in them adapting fast to various conditions and developing resistance, whether it be to fungicides or to cultivars. If we look at the two different Alternaria species, Alternaria solani infects senescing or older tissue. Young plants and plant tissue of potatoes have an inherent resistance to Alternaria solani. So it's very, very seldom or unlikely that you will see early blight symptoms on young plant tissue. Alternaria alternata, on the other hand, can infect younger tissues as the potato plant does not have any inherent resistance to Alternaria alternata infection. Looking at the symptoms of the two diseases, starting with early blight. Early blight is seen by dark brown to black necrotic lesions that are found on leaves and on stems. And the characteristic symptoms of early blight are concentric rings in the lesions. These are due to alternating day or night temperatures and moisture conditions. So when one sees these very characteristic target spot lesions or concentric rings, it is a characteristic and almost diagnostic symptom of early blight on potatoes. What we also often see is a chlorotic halo around the lesions, and this is due to a toxin that is produced by Alternaria solani, which results in chlorosis of the tissues surrounding the necrotic areas. Lesions often start as small lesions or remain small under unfavorable conditions. They are usually oval, but they can become large coalesced lesions, as you can see in this middle photo in the top, under favorable conditions. So it is quite often that one can see a completely blighted leaf under favorable conditions. Symptoms on tubers appear as dark, sunken lesions on the tubers, although it is not that common to see Alternaria solani infections on tubers. Tuber infections occur when there's a very, very high inoculum pressure in the field. Brown spot or molrus, on the other hand, is characterized by the first symptoms being seen on the underside of leaves. So because of the fact that the symptoms are first seen on the underside of leaves, it is critically important that one turns the leaves over when looking for brown spot. The lesions are dark black to brown. They're small, round necrotic lesions on leaves and on stems, as you can see in these pictures. And a very distinctive property that distinguishes brown spot from Alternaria solani, in other words, early blight, is that there are very few to no concentric rings in the lesions. On tubers, the symptoms look almost identical to those of Alternaria solani, and there are dark sunken lesions on tubers. Just to point out to you, there are other symptoms that appear very similar to either or both early blight and brown spot. The first is a manganese deficiency. In this case, brown flecks are seen, and they're normally observed along the leaf veins or on the outer edges of young leaves. So one would have a look at the pattern of how the lesions are distributed on the leaves to make some sort of a diagnosis as to whether it's manganese or brown spot. Tomato spotted wilt virus, or Tomaticromnac virus, shows similar symptoms to Alternaria solani, to early blight, in that there are sometimes concentric rings in the lesions. However, the lesions are normally smaller than those of early blight, and they are usually grouped, as you can see in this photo. Another characteristic diagnostic property of tomato spotted wilt is that infected plants can be found next to healthy plants, and this is because the virus is spread by seed. Then ozone damage, although not seen that often, can cause interveinal browning of leaves, and this is sometimes also confused with one of the two Alternaria diseases. We're going to move on to the disease cycle now, and I'm going to take you through the disease cycle, explaining the infection process, the conditions that are required for infection, and then ending back with initial inoculum. So any disease will start with primary or initial inoculum. In the case of the Alternaria species, Conidia or Mycelium that have overwintered in plant debris, on seeds or on tubers, serve as primary inoculum. This primary inoculum can survive between 5 to 8 months on plant debris or in the soil. It's been noticed that the longest survival is actually in dry, fallow soil. And this, of course, we find a lot in South African conditions. So how is this inoculum disseminated? Well, primary inoculum is primarily disseminated by wind and by water to other plants in the field. Secondary inoculum that one can see here on the right-hand side of the slide is also disseminated by water, by wind, but also passively by insects such as aphids, where they actually carry the spores on their mouth parts or on their legs or on their bodies. So now we move on to, once the primary inoculum, or secondary inoculum for that matter, arrives at a plant, the spores need to start germinating. So for spore germination of Alternaria solani, free water or high relative humidity is required. Temperatures above 20 degrees C are required for spore germination of Alternaria solani. For infection of the plant, remember I said that Alternaria solani does not require wounds or natural openings, although it can use them to infect. Alternaria solani can penetrate the plant directly. Infection occurs when there's free water or high relative humidity again, and the temperatures are between 10 and 35 degrees C. The optimum temperatures for infection by Alternaria solani are between 20 and 30 degrees. The picture for Alternaria alternata is relatively similar, but there are slight differences. Spore germination also occurs when there's free water or high relative humidity, and the temperatures are above 20. The optimum temperature for spore germination for Alternaria alternata is approximately 25 degrees. You'll recall that I said Alternaria alternata cannot penetrate plants directly. It requires penetration through wounds or through natural openings. For infection, the primary difference here is that there is a wider range of temperatures at which Alternaria alternata can infect, up to 40 degrees and a little bit higher, and the optimum is between 20 and 30 degrees C. Again, free water or high relative humidity are required. For sporulation of both Alternaria solani and Alternaria alternata, temperatures between 5 and 30 degrees are necessary. So that's a very wide range of temperatures. The optimum is around 20. Most sporulation occurs after a heavy rainfall event, irrigation, or dew. So one needs to be aware that after rainfall, it is likely that you'll get sporulation and then the spores will start dispersing. And so this is when one needs to be on the lookout for new infections in a couple of days. Most spores are produced when the conditions consist of alternating wet and dry conditions, particularly when the wet conditions occur at night and then they are followed by dry days. So this would occur if there's high relative humidity and a lot of dew at night, or perhaps if irrigation occurs at night and then the plant surfaces dry out again through the day. This is when one will get most spores being produced. So we want to try and avoid this if at all possible. So taking these factors into account, how do we manage these diseases? Well, firstly, because we know that the pathogen can survive in the soil, we want to practice a 3 to 5 year crop rotation with non-hosts. Now, this doesn't only apply for the two Alternaria species. This is a very sound management practice for any pathogen and, in fact, for any host. It is also important that we try to increase the plant's resistance, and this can be done through numerous biological products that are available. Biological products can also be used to actually manage the pathogen itself, to provide competition or to enhance the plant's resistance to the pathogen. There are numerous products that have been used with a fair amount of success. A fungicide program with registered products can be followed, and I'll discuss this in the next slide. It is important to do irrigation scheduling to ensure optimal water management. If plants remain wet for too long, it will increase the chances of infection. And then it's also important that the plants have optimal nutrition, not too much and not too little. One should try to aim to increase the nitrogen up to a certain point, increase potassium, and lower phosphorus. Good insect and weed control is important. I've mentioned that weeds can passively transmit spores or transfer spores from one plant to another, and weeds often serve as alternative hosts for the pathogen. So we want to remove all weeds from the area. In this way, we can try and ensure healthy plants. So just something quickly about a fungicide program. One should always follow a registered program. And normally what the standard practice is to follow a registered program for early blight, because there are more fungicides registered for early blight than there are for brown spot. If one has a problem with brown spot in the season or historically, it is wise to start the spray program earlier in the season than you would for early blight, and to start with contact fungicides. What is critically important in a fungicide program, any fungicide program, is to alternate active ingredients from different frac groups. This lowers the chance of resistance development in the pathogen population. It extends the lifespan of the active ingredient, and it ensures sustainable management of the disease. We need to bear in mind that there is already reduced sensitivity to the QRI fungicides in the South African Alternaria alternata population, and there are indications that there may be reduced sensitivity to other active ingredients. It's been documented in many parts of the world that Alternaria alternata develops resistance or reduced sensitivity very quickly to numerous active ingredients in different frac groups. So it is important to alternate active ingredients from different frac groups. What is also important to ensure sustainable disease management, and prevent development of resistance is to combine and or alternate contact and systemic fungicides in the spray program. So when we put this all together, I like to put it back into the disease pyramid. If you have ever had a lecture from me as an undergrad or in any of my other courses, you will know that I always bring things back to the disease pyramid. For disease to develop, we have to have a virulent pathogen, a susceptible host, favorable environment for disease to develop, and when these three are present, we have the disease triangle in place. If they are present, disease can develop. But we must not forget that man can play a significant role in minimizing one of these three factors. What's also important is the time that these three factors have to interact. The longer a virulent pathogen is in contact with a susceptible host under favorable environmental conditions, the more disease will develop. So we need to aim to reduce contact time and to reduce favorable environmental conditions in order to reduce disease in the end. Thank you for your time.